All right, in this lesson we're going to look at regulating how genes are used and when genes are used by cells. So we're looking at how it is that a cell can turn genes on and off so that you don't have excess cell products building up, excess proteins building up that aren't needed by the cell. The first way that you can see cells regulating their genes is using what we call an operon system. Okay, and you can see this described here. These are used primarily by bacteria, and essentially an operon you can think of as kind of like an on-off switch for a particular gene. So ideally what happens with bacteria is when lactose enters the cell, it needs to be broken down by this enzyme, beta-galactosidase, to be broken down into glucose and galactose. The problem is you don't really need beta-galactosidase most of the time. And so there's no need for the cell to constantly have that gene on. Really, only if lactose is present do you need the gene to be produced. And so what happens is essentially genes that control the regulation of beta-galactosidase are packaged into these operons. And oftentimes you would have several related genes together within the same operon. And then there's one on-off switch as part of that operon. So to regulate the operon, what you have is a promoter region. So this is an example of the LAC operon that produces the beta-galactosidase gene. And in the LAC operon, you have three genes, LAC-Z, LAC-Y, and LAC-4. Or sorry, LAC-A. And then you have this promoter region and this operator region. Okay. Down here is a regulatory gene just upstream from the operon itself that can produce the repressor molecule. Okay, so essentially the repressor molecule is able to clamp down onto this region here, the promoter region, and if it's blocked, if that promoter region, or sorry, the operator region, if the operator region is blocked, then this promoter region where the RNA polymerase would normally attach to try to make these particular genes, it would attach here, but if this region is blocked, then transcription can't proceed and the gene is effectively turned off. Okay, and there are actually two types of operons. There are operons that are inducible, meaning that the operator is generally, the default position is that this operator is generally blocked by some sort of repressor molecule. And then there are repressible operons, which the default position is on. So you would normally not see anything bound to the operator, and this gene would normally be open all the time. So let's look at what this looks like. So this is the LAC operon itself. Okay, so this LAC-I gene is producing the repressor molecule, and the repressor molecule clamps down on this operator right here. And it's normally on the operator, and here's our RNA polymerase. It's trying to transcribe these genes, but because the operator is blocked, it's not able to move downstream and get to these genes that we're looking at, LAC-Z, LAC-Y, and LAC-A at the end. However, when lactose is present, what you can see is the lactose molecule binds to the repressor. It changes the shape of the repressor so that the repressor falls off. Okay, and therefore now this RNA polymerase is able to move downstream and transcribe the DNA so that you can get an mRNA molecule, you can create that beta-galactosidase. If you don't have a functional repressor gene upstream, then essentially this operon would be on all the time. But again, this is what we call an inducible operon. You have to induce the operon to turn on. Okay, its default position is off because the default position is to have this repressor molecule bound unless lactose is present. Okay. By contrast, the trip operon is what we call a repressible operon. All right, most of the time cells need tryptophan. Okay, most of the time, and so tryptophan is always being produced. So most of the time, tryptophan is absent, and therefore the repressor molecule is not bound to our operator, and therefore transcription just continues on. So you're constantly making tryptophan in cells, or the enzymes that are involved in tryptophan synthesis. But when tryptophan starts to build up, tryptophan attaches to the repressor molecule, then that activates that repressor. Okay, that changes the shape of the repressor such that now it's capable of binding to the operator. And when that happens, it turns the gene off. So here, the default position is on. That means this operon is what we call repressible. And you can see the two operons summarized right here and compared to one another. Here, this is an inducible operon. This one is an inducible and repressible operon. All right, let's look at how does a eukaryote regulate its genes. You might have a few operons in eukaryotes, but primarily where eukaryotes are concerned, there's a bunch of different steps. These steps exist in prokaryotes as well, but remember, prokaryotes lack that nucleus, and a lot of the processes that are used in prokaryotes are coupled. Remember that? So you end up having transcription and translation happening at the same time. It's hard to regulate transcription when you don't, that's coupled with translation, so translation's going on at the same time as transcription. So eukaryotes are a little different. This nuclear envelope provides a nice barrier that allows us to take a pause in between transcription and translation. But notice there's also a bunch of different steps, if you're looking at this picture, where you can regulate in some way the molecules and prevent them from opening up, or keep them closed, or you can turn them on, turn them off, and so on. And so there's like a whole bunch of different ways here. You can regulate the level of the chromosome, you can regulate transcription, you can regulate RNA processing, you can regulate the stability, the amount of time that the mRNA sticks around in the cytoplasm, and you can even regulate translation or even post-translation. So there's a whole bunch of steps where we can stop, pause, do something to our molecules to turn them on and activate them, or to turn them off. So really, bacteria only regulate transcription, and they do so using operons. Eukaryotes have a lot of different mechanisms for doing that. All right, the first mechanism is DNA regulation. And in one of the previous videos, I talked to you about what chromatin is, and I mentioned that it's really important to understand that. You have DNA packaged in what we call nucleosomes, right? This is a nucleosome. It's a piece of DNA that's wrapped around 8 histone proteins, right? And then it's strung along to the next nucleosome and to the next, and so on. This is what you're seeing in this electron micrograph picture here. This is chromatin. Okay, so chromatin is in this string of pearls configuration that you see, where you've got the string and then you've got the nucleosome that looks like little pearls on a string. Okay. And so under normal conditions, the histones have these lysine tails that extend out from the nucleosome. So they would kind of extend out like this. They're used to kind of attach to other nucleosomes, right? If you add an acetyl group to those tails, what happens is these histones don't tend to coil up, they tend to push apart from one another. So this is what acetylated histones would look like, right? Where you can see that this is all open, the histones are pushed apart. When you take those acetyl groups off, what happens is these histones become attracted to one another, and they start to coil into this structure, into this condensed structure that you see here. Now, it makes sense then that when the DNA is all open, when the nucleosomes are all separated apart from one another, and the chromatin is not condensed, it's open for business. It's available for enzymes to attach and for transcription to take place, right? When it's condensed like this, just by removing those acetyl tails, then what happens is the DNA condenses up and you really can't access this portion of the DNA molecule. Another thing that you can do is add methyl groups. You can attach methyl groups to cytosine, which also will cause the DNA to coil itself up. Okay, and so methylated DNA will look more like this, or like this. There's the condensed chromatin as well. So what happens is that some research shows that some heavily methylated areas will recruit deacetylation enzymes as well. So if we if we methylate the DNA, what happens also is enzymes get attracted to that area to also pull off the acetyl groups from the histones, which causes condensation. And again, you can see that here. Okay, here's decondensed chromatin, here's condensed chromatin that you are seeing there. These patterns of different areas that are methylated, different areas that are acetylated, this is what we refer to as the epigenome, the on the genome. So it's not just what genes do you have, it's what's on top of your genome causing it to either open up in some places or condense down in other places. And it turns out, and this is kind of brand new science that we've just started to realize is a big deal, but it turns out a lot of that epigenetic stuff is inherited from your parents, grandparents, and so on. So which parts of your genome end up methylating and acetylating and deacetylating and so on is an inherited factor. So it turns out these nucleic acid things that are on the genome also regulate your genome. All right, now in terms of regulating transcription, right, there's a couple different things that can happen here. Transcription factors bind to the DNA and make it easier for RNA polymerase to bind to the DNA. So if you put specific transcription factors and also enhancers, if you add these transcription factors to a gene right at a promoter region, and then you have enhancers up and downstream from that gene, those enhancers actually tend to activate transcription, and you saw this in the video. You saw in the transcription video where you had everything attached. You had the factors promoted here, those at the promoter. Those factors recruit the RNA polymerase to come along, and then when the enhancers activate everything, it caused transcription to happen in the video. So, attaching transcription factors to the head of a gene can help you to ensure that transcription is going to take place, and then these distal control elements as well, these enhancers can also initiate or ensure that transcription of that particular gene is going to happen. So if you don't put the promoters on, if there aren't enhancers present, then it's a little less certain that transcription of those genes are going to take place. But if you do have them, then it ensures that transcription is going to take place. And then I mentioned this, there are some co-expressed genes in eukaryotes. So but oftentimes each gene has its own promoter. So typically, instead of using an operon system, this is how eukaryotes are going to regulate genes. All right, after that, there's a couple steps that you saw from the other slide about how you can regulate genes and regulate how they're turned on or off. The last video mentioned introns and exons, right? And I mentioned that there is some genetic overlap, that you can actually alternatively splice a gene, that primary transcript that comes off of the DNA molecule. Splicing it in one particular way might cause it to form one kind of protein, but if you splice it in a different way, it might form a different protein. So that's one thing that can happen, the way that you process the RNA molecule, that primary transcript. RNA degradation, and this is something we're combating right now in the age of coronavirus. People would love to do mRNA vaccines, essentially, where instead of injecting you with pieces of the coronavirus, they would like to inject you with mRNA from the coronavirus so that your cells make viral proteins and then your immune system reacts to them. The problem is mRNA is fleeting. It doesn't hang around very long, usually, and they're not really sure how well that's going to work because they're not really sure how long they can get that mRNA to stick around. Sometimes eukaryote mRNA can last a little bit longer than what you would see in prokaryotes. But sometimes, you can have enzymes that are just patrolling cells looking for mRNA that's just kind of floating around in the cytoplasm and waiting to break it down, because essentially what the cell needs to do is recycle those nucleotides back to the nucleus so that more RNA can be made. There's also initiating translation. You can block the 5' end of the mRNA, so you can actually synthesize a protein that would block it, which means now the mRNA can't attach to the ribosome until we take that particular protein off. Sometimes when translation happens, or transcription happens, the poly A tail is not long enough, and so you've got the RNA waiting, it's ready to go, but then it needs more adenines attached to it before it's long enough for protein synthesis. And then also there's protein processing and also protein degradation that happens. So some proteins require post-translational modification, like reversible phosphorylation, like you saw in a signal transduction pathway where you phosphorylate something, right? So sometimes proteins aren't active until they're phosphorylated. Sometimes you get extra pieces of amino acids attached to the end of a protein so that it's not active until you come along and clip those off. That's particularly useful for things like digestive enzymes that could really cause some damage inside of cells. And then some proteins get tagged with something called ubiquitin, which alerts enzymes that break up proteins to the fact that they're there, and those enzymes will be recruited toward that ubiquitin and will degrade those proteins. And again, if you've got some proteins or enzymes that are no longer needed, it's helpful to go ahead and break those down so that you can recycle the amino acids into new proteins. So all of those things are steps that can be taken to regulate genes, regulate the RNA that you get, regulate translation, and then also regulate the proteins that are produced so that cells have complete and utter control over which genes are being turned on and off and which genes are being used at any given time.